Good afternoon, everybody. Thank you all for coming today, and I'd also like to extend a welcome to our guests who are not in this room right now, but are watching via Skype. Today gives me great pleasure to introduce Erica Bakota for her thesis defense today. Erica actually did her undergraduate degree here at Rice, which is where I had my first opportunity to meet her as one of the TAs for sophomore organic chemistry. After she finished her degree here at chemistry, she decided that she wanted to go and visit California, and so she spent about a semester in California in a program in food science, and a PhD program in food science. And after that semester, she decided that she needed to come back and do, I suppose, more fundamental work in chemistry. And so it was with great pleasure that we welcomed her back to the Rice campus, where she started work in my lab January 2007. Erica has shown from really right away, actually before she even joined my lab, that she can be a proficient scientist. She did undergraduate research with Bob Hauge and Rick Smalley. Where together they resulted in 4 publications. Since coming to my lab in 2007, she has an additional 6 publications, and we have 1 more publication that we expect to submit before she can leave. Today she's going to tell you about that work that's been done over the last 4.5 years approximately. So it gives me great pleasure to introduce Erica, where she'll be talking about multidomain peptides, sequence-nanostructure relationships, and biological applications.
Well, thank you, Jeff, and thank you all for coming today. My name is Erica Bakota, and I'll be talking to you today about multidomain peptides. Now, the work I'm going to talk about today primarily draws from 6 publications that are either in press or have already been published, the first of which appeared in the Journal of the American Chemical Society in 2007, and that introduces the multidomain peptide series. From there, I'll talk about some covalent modifications that we can make to multidomain peptides, and then after that, I'll cover 2 applications of multidomain peptides. The first is to use these peptides as surfactants for single-walled carbon nanotubes. The second application I'll talk about is to use multidomain peptide hydrogels as a method of drug delivery. Now, first and foremost, the Hartger lab is a self-assembly lab, which is to say that we utilize non-covalent interactions between distinct molecules to direct the formation of larger and more complicated structures. And the key here is that these structures have controlled size and shape. And we do this with peptides. And so each of these pictures you see here, we've been able to make using multidomain peptides and replicate some novel structures at the nano level. Now, we do this primarily by solid phase peptide synthesis, and this uses an insoluble polystyrene resin onto which an amino acid is loaded. It's protected with an FMOC group. We can deprotect that to expose the free amine, add an amino acid, which is also FMOC protected, and form a peptide bond. Now, we can deprotect that FMOC group, exposing the free amine again, add a second amino acid, form another peptide bond, and so on. And this is how we grow the peptide chain on the resin. So with that, I'd like to introduce multidomain peptides. And these are peptides that are designed to self-assemble into nanofibers. And as the name suggests, they have several domains. We have a hydrophilic domain, shown here in purple, made up of glutamine or serine residues that interacts with water. We have a hydrophobic domain, shown here in green, and that can be made up of leucine or phenylalanine residues, and that is shielded from water in the nanofiber. And then we have charged residues that make up the final domain, composed of lysine or glutamic acid, and these repel the peptide strands from one another and discourage self-assembly. Now, when we put all these domains together, we can abbreviate this peptide as a large color block, as you see here. And when we put 2 of those blocks together, we get a peptide dimer in which the hydrophobic or water-hating portion of the peptide is sandwiched in the middle, and the hydrophilic portions of the peptide are on the outside interacting with water. We also have the charge domains on the end, and when we actually add salts into this solution, the salts will screen the charges on the charge domains and take away the disassembly force, and we actually get self-assembly into nanofibers. And when the nanofiber network is allowed to grow and develop, we actually end up with a macroscopic hydrogel. Now, the first series of multi-domain peptides that we looked at followed the KQL motif, so lysine as the charge residue and QL repeats in the central block. And what we found was that we actually had to strike a balance between having too many charged residues and too much charge repulsion, and an insufficient central block in the center of the peptide. So you can see that K2QL3K2 does not form nanofibers, nor does K7QL6K7, but these three peptides in the middle, K3QL6K3, K2QL6K2, and K2QL5K2, all do form nanofibers, the best of which is K2QL6K2, and I'll talk a little bit more about that now. Now, when we make a peptide, we need to characterize the secondary structure of the peptide, and we do that using circular dichroism, or CD, and infrared spectroscopy, or IR. Now, in a CD of a peptide that self-assembles into beta sheets, one thing that we are looking for in the CD spectrum are two features that tell us that indeed we have assembled into a beta sheet. The first of which is a maximum between 190 and 200 nanometers, and a minimum around 216 nanometers. Things we're looking for in an IR spectrum are amide 1 band, which is centered around 1625 wave numbers, and an amide 2 band centered around 1525. These four features in the CD and IR tell us that we have in fact made a beta sheet, which is the secondary structure that allows these peptides to self-assemble into nanofibers. We also characterize our peptides by microscopy. On the left, you see a cryo-TEM image of the nanofibers made by K2QL6K2. Now, we do cryo-TEM on all of our peptide solutions because these peptides are quite prone to aggregation, and doing cryo-TEM as opposed to regular TEM allows us to avoid the drying artifacts that occur when we do dry TEM on peptide structures. And so we can see that in the solution state, without any charge screening salts, we are forming nanofibers. And then when we actually add the salts and make the gel, we can image the gel by doing critical point drying on that gel, and then imaging by SEM. And in the gel state, we actually also see a nice fibrous network. The last way we characterize a peptide is to do rheology on the peptide hydrogel. Rheology is basically looking at the mechanical properties of the gel. And in this test, which is a strain sweep, we're looking at two parameters. We're looking at G prime, which is the elastic modulus, or how solid-like our peptide gel is. And then we're looking at G double prime, which is our loss modulus, and that's telling us how liquid-like our peptide gel is. Now, in a good peptide gel, we're going to see a G prime that's at least an order of magnitude greater than the G double prime, and we're also going to see a drop-off point at some level of strain. And so what this plot tells us is that we have, in fact, formed a good hydrogel with a G prime of about 95 pascals. We did one more characterization on this peptide, K2QL6K2, and that was to look at it by AFM. And so we looked at AFM of the dilute peptide solution. I was able to see short nanofibers, measured the heights on those nanofibers, and those turned out to be between 1.9 and 2.0 nanometers. That actually corresponded exactly to our physical model, which predicted a height of 2 nanometers. And so through these characterizations, both spectroscopically, rheologically, and also microscopically, we were able to say that this peptide is indeed self-assembling in the manner that we think it is. Now, given that, is it possible to tailor multi-domain peptides for specific applications? Well, to answer that question, we have to know what specific applications we might use them for, or what it is that we want to get out of this peptide system. One of the primary things we'd like to get is better rheological properties. We would also like to demonstrate tunable fiber morphology. And then if we're going to use these peptides in a biological setting, we'd like to demonstrate both biocompatibility and biodegradability. And towards those four ends, we made a number of substitutions. The first of which is a substitution in the hydrophobic block of the peptide, and that's a leucine to phenylalanine substitution. We also made a leucine to tryptophan substitution, and both of those substitutions result in the introduction of pi-pi stacking into this system, and we think that pi-pi stacking may be able to direct the self-assembly process to give us different fiber morphologies. We also made a substitution to the hydrophilic block of the peptide, substituting glutamine with serine, and this resulted in peptides that have a stronger tendency to form beta sheets, and ultimately they have better rheological properties. We made one more substitution to this template, and that was to replace lysine residues with glutamic acid residues in the charged block of the peptide, which resulted in peptides that ultimately were more biocompatible. And we'll talk a little bit more about that. Now, the first substitution we made was a leucine to phenylalanine substitution. You can see in the CD, we do in fact have our maximum between 190 and 200 nanometers. We have our minimum around 216. And we also have this new artifact that has cropped up around 202. And while this may look like a significant deviation from a standard beta sheet CD spectrum, it has been widely reported in the literature actually that phenylalanine-phenylalanine interactions within peptides can create this artifact. And so what we are actually looking at here is a beta sheet secondary structure. If we look at the IR, that confirms that this is actually a beta sheet. We see a clear amide 1 and amide 2 band. But if we look at this by cryoteum, we actually don't see the same nanofibers that we saw with K2QL6, K2. So we see the formation of micelle-like structures. However, when we do add the charge screening salts to this, we do see branched nanofibers in the gel state. If we do a rheological characterization of this peptide, we see that we do actually form a nice peptide hydrogel with a G prime of around 200 pascals. So the absence of nanofibers in the cryo-TEM images is not necessarily indicative of its inability to form a gel. Now when we look at K2QW6K2, we see the return of a more traditional looking beta sheet spectrum by CD. We see a beta sheet spectrum by IR. And we see the return of long, linear, and uniform nanofibers in the solution state. Now, also if we look at this by SEM, we see that there are indeed long nanofibers there as well. However, when we look at the rheology of this peptide, it turns out that it doesn't really form a gel at all. So a G prime of about 20. Now, this really isn't a whole lot better than water. And so while we are forming nanofibers, that seems to be unconnected at this point from our ability to form a gel. Now I want to move on to a different substitution, a Q to S substitution. So when we incorporate serine into this peptide, we see in K2SL6K2, we have a traditional beta sheet spectrum by CE and IR. And we have a very nice nanofiber network by both cryo-TEM and also by SEM. Now when we do a strain sweep on this peptide gel, we see a G prime which is the best yet around 300 pascals, which indicates that this Q to S substitution has actually done a lot for us in terms of improving our rheological properties. And so this is something that we'd like to work with a little more in the future. The last peptide I want to introduce to you incorporates 3 of these modifications at once. E2SL6, E2GRGDS has an SL repeat in the middle block of the peptide. So instead of QL, we now have SL. Instead of lysine residues on the termini, we have glutamic acid residues. And finally, we've incorporated the RGD sequence, which is a known cell adhesion sequence. And cells in the body will naturally grab onto this RGD sequence and anchor to it. And so we think that that can improve the biocompatibility of this peptide. So we see that it does form a beta sheet by CD. We have our traditional maximum and minimum here, and we see our amide 1 and amide 2 bands by IR. And when we look at this by AFM and SEM, we see perhaps the best nanofibers network that we've seen yet. So you can see that even by AFM, which is a drying technique, we see long, linear, and uniform fibers. When we look at this by SEM, you can see a very strain sweep on this peptide, we see a G prime of about 475 pascals, which means that these modifications have led to a peptide that is very appropriate for a number of biological applications, and I'll talk a little bit more about that in a little bit. But just to summarize this section on sequence structure relationships, there are a number of modifications that we can make to improve the properties of our peptides. The first of which is to change lysine to glutamic acid. The second is to change leucine to an aromatic residue such as phenylalanine or tryptophan. We can change glutamine to serine in the hydrophilic block of the peptide, and we can also add bioactive sequences such as RGD, or something that I'm not going to cover very much is this enzyme cleavable sequence SLRG to enhance biodegradability in vivo. Now, after this, we're left with the question, are there any other modifications that we can make outside of this set of parameters, so things that are not sequence modifications, that can improve the properties of these peptides? And the answer to that is yes. And normally when people talk about improving the rheological properties of a peptide gel, people think of introducing cross-linking into the system. And in proteins, traditionally, that's done by introducing disulfide bonds. That means incorporating cysteine residues into your peptide. However, after synthesizing quite a few of these, we found that working with cysteines is a little bit difficult because it's hard to control the oxidation state of cysteines. And so we were looking for another method that allowed us more precise control over the cross-linking state. And so what we found is we were actually able to induce cross-linking between multi-domain peptides using either plasmamine oxidase or lysyl oxidase, which both cross-link using lysine residues rather than cysteine. We were able to form those cross-links either between lysines on the same nanofiber and also on different nanofibers. Now, the way that we stumbled upon this in the first place is we're taking a peptide K2SL6K2 and adding it to culture with dental stem cells. And what we found was over the course of about 2 weeks, the G prime of peptide hydrogels incubated with dental stem cells increased dramatically. And so this blue set of bars here is with cells. And so we can see that we're reaching G primes in excess of 6,000 pascals after 2 weeks. And we thought, well, maybe this is an effect that comes from the cells. Maybe they're secreting extracellular matrix or something like that. And so what we did is we took the cells out. We still had identical cell culture conditions, but we took the cells out and we saw actually the exact same increase in G prime as we did when we did have cells. So there's something about the cell culture condition that is increasing our G prime in the system. And so what we did is to systematically go back and subtract various aspects of the cell culture medium. And we saw that if we incubated with only PBS, we did not see an increase. And then when we add in basically anything with serum in it, FBS is fetal bovine serum, we see an enormous increase in G prime. And when we incubate in serum only, we see something that's even better than what we see from the cell culture conditions. And so we could say that it actually was the serum that was cross-linking our peptide. Now, thinking that it may be possibly was lysyl oxidase, which is a known component of the serum, we took an inhibitor to lysyl oxidase and added it to the cell culture conditions. So this is beta-aminopropionitrile here, and saw that the increase in G prime was not as great when we inhibited the enzyme. So that narrowed it down basically to lysyl oxidase. Unfortunately, lysyl oxidase is not commercially available, so we went with the next best thing, which is plasmamine oxidase, and added that to samples of K2SL6K2 hydrogels without anything else. So just plasmamine oxidase and hydrogel, and we were able to observe over 15 days a huge increase in G prime of those gels. We also looked at this by AFM, and at day 0, before we add the enzyme, we see that we have a somewhat uniform nanofiber network, but after 7 days of treatment with the enzyme, we see highly bundled and highly cross-linked fibers, which has indicated that the plasmamine oxidase did in fact cross-link this system. to our big picture. So we can make all of these modifications to improve the properties of our peptides, but we can also do things that are not sequence modifications. We can also make different covalent modifications to our system by using enzymes. So with that, I want to talk a little bit about some applications. The first one is to use multidomain peptides as single-walled nanotube surfactants. And this is a project that we've done in collaboration with the Weizmann Lab, and specifically with Demon Cycle C. As you all probably know, single-walled carbon nanotubes are an allotrope of carbon. They have unique optical and electrical properties that make them very attractive for applications in medicine, sensing, materials, and electronics. However, their solubility in aqueous systems is a huge problem. And so if we're going to use nanotubes for practically any biological application, it's essential that we bring them into solution using an appropriate surfactant. So the current approaches we have to deal with that problem are several. The first is covalent modification, which does result in good solubilization of tubes. However, when we destroy the sp2 nature on the surface of the tube, that actually destroys the fluorescence, which is what makes nanotubes so attractive to use in the first place. And so covalent modification is not appropriate. We also have our standard ionic surfactants such as SDS and SDBS, which actually allow good fluorescence from the tube, but are known to be highly cytotoxic. And so if we want to use these for a biological application, we have to go with something that's a little more non-toxic. So people have already figured this out and come up with a biocompatible class of surfactants, which are relatively non-toxic. However, because the interaction between these surfactants and the tubes are non-specific, so these are polymer-based surfactants that have sort of a messy coating around the tubes, the fluorescence that they allow from the nanotubes is relatively weak. And so what we would like to do is preserve both the fluorescence and have good solubility and be non-toxic. And we think that peptides can actually satisfy all three of these conditions. But that begs the question of why peptides and why not some other molecule that's not one of these first three categories? Well, the answer is that peptides offer a way to wrap the tubes without any kind of covalent modification. By engineering a specific interaction between the surfactant and the nanotube, we actually have the possibility of tuning the optical and electrical properties of the tube. Because peptides are biological in nature, their potential for biocompatibility is quite high. And with our system, we can actually incorporate a biotargeting ability by adding sequences such as RGD to these peptides to direct what they do in vivo. So this is the way it works with multidomain peptides. We have our peptide dimer here made from K2QL4K2. We can approximate that with these red blocks. The blue squiggly lines are our lysine residues. We're going to further approximate that as a sandwich. And then we add our nanotube, and that nestles into the center of the sandwich. So the hydrophobic surface of the tube interacts quite nicely with the hydrophobic portions of the peptide. And in fact, we've completely wrapped the tube to make a very delicious nanotube hot dog. Now, when we test our initial series of multidomain peptides as nanotube surfactants, we find something interesting. So, as I said before, these three peptides, K3QL6K3, K2QL6K2, and K2QL5K2, all form beta sheets, and they're all capable of forming nanofibers. It turns out that these are the same peptides that are capable of solubilizing nanotubes. And so what we think is happening is the nanotube is actually acting as a template for self-assembly and allowing nanofiber formation around the tube. And the best of these happens to be K2QL5K2, but we're going to take a closer look at all three. These are E, F, and G in our series. So if we look at E, F, and G, we see that all of these allow decent fluorescence from the nanotubes. And if we look at the absorbance, which is basically a measure of how much nanotube material have we suspended in solution, we see that E, F, and G all perform much better than all the rest of the peptides, with the best of these being, again, K2QL5K2. That said, we thought we might be able to do a little bit better. So we know that this KQL model suspends nanotubes with better fluorescence than fluoronic. However, there are a variety of amino acids available to us. So we have all the functional groups in the world available for us to incorporate into our peptides, and we think that amino acid substitution to these peptides can yield even better fluorescence from the nanotubes. And so we made the same modifications that I talked about earlier. We made a glutamine serine modification, which results in a peptide that has a stronger beta sheet forming tendency. However, I'm not actually going to talk about that because it failed miserably. What happened was we made a peptide that was so good at forming a beta sheet that it was only interested in forming nanofibers with itself and not particularly interested in forming any kind of interactions with nanotubes. And so these SL peptides turned out to not solubilize tubes hardly at all. However, we made a few other modifications that did work a lot better. We made a leucine to phenylalanine modification and a leucine to tryptophan modification. This leucine to phenylalanine modification resulted in better fluorescence from the tube, and we think that pi-pi stacking is possibly playing a role here. We also made a lysine to glutamic acid modification to make these peptides more biocompatible. And so if we just look at a handful of these peptides, these all incorporate the lysine to glutamic acid modification. They do allow good fluorescence from the nanotubes. And then if we look at the absorbance, so how much material have we suspended, we can see that we have suspended a fair amount of material because we see distinct Van Hoek transitions in the absorbance spectrum. If we add K2QF5K2, the fluorescence and absorbance improve. And if we add K2QF6K2, things improve even more. And so we said here, okay, we're on the right track. We are suspending a bunch of material. It's allowing good fluorescence from the tube, and it's a peptide, so let's go ahead and introduce this into cell culture. Before we did that, we had to figure out exactly which one of these we wanted to use. And so where these peptides really shine, this new generation of peptides, is when we look at the fluorescence to absorbance ratio. And that allows us to figure out the reason why we're getting good fluorescence out of these samples. So are we getting good fluorescence because these peptides have a specific interaction with the tube and it's allowing good fluorescence from the tube? Or are we getting good fluorescence from the sample because we've just suspended a whole bunch of material and because we have so much stuff there, we're getting a higher overall signal? Well, it turns out if we normalize for absorbance, the peptides with 5 repeats in the central block perform the best, K2QF5K2 and K2QL5K2. If we take the second one, K2QL5K2, and add it to cell culture, we get this disaster on the left. So what has happened here is immediately on contact with the cell culture medium, this nanotube suspension in K2QL5K2 has interacted with the phosphate ions that are naturally present in cell culture medium. Those phosphate ions have screened the charges on the peptide, and we've immediately gotten a big, nasty chunk of nanotube gel. And if we watch this with PBS, it stays put. These nanotubes are not going anywhere. And so that says to us that peptides with lysines are actually really not very compatible with cell culture at all. And so we needed to come up with a peptide that could effectively deliver tubes to cell culture without aggregating on contact. And to do that, we went back to our peptides with glutamic acids on the end, and we were able to suspend nanotubes, and when we added that to cell culture, we saw that actually it dispersed quite nicely. So if you didn't know any better, you would say there's actually not anything that we've added to the solution here because everything has been so completely dispersed. Now, just because we've done that, this does not actually mean we've effectively delivered nanotubes to cells, and it doesn't necessarily say anything about the cytotoxicity of the peptide surfactants. So we decided to do a cytotoxicity study. And these here are NIH3T3 fibroblasts. And these have been incubated with a variety of different surfactants. So we've taken one of our standard ionic surfactants, SDBS, incubated that with cells, as well as floronic, and also 2 peptides, EQL6E and EQL6E-GRGDS. So that has that cell adhesion sequence incorporated in there. We see that when we incubate these cells with SDBS, we get total cell death. So these cells are rounded and damaged, and this is not a healthy cell population at all. In a healthy cell population, what you will see is you'll see a combination of cells that are anchored to the bottom of the well plate, like this one, or also dividing, like these cells. And so you'll see some rounded and some anchored. And so basically what we're looking at with chloronic F68, EQL6E, and EQL6E GRGDS, that by all appearances we have a healthy cell population. Now, that says, okay, they're probably not dying immediately. We don't actually know what's going on with the surfactants or the nanotubes by just looking at these images. So we set up some experiments with a fluorescence microscope that was actually built in the Weissman lab. And this was an experiment done with Dima Cybulski. And what we did is we actually took these cultures that had the surfactant suspended nanotubes added to them, and we were able to image the nanotubes inside of the cells. So this microscope detects fluorescence in the near IR range. And so what we're actually looking at is a signal from nanotubes here. This is the outline of the cell body. You probably can't see it very well. But what this looks like is we have nanotubes inside of the cell. To figure out whether they were on the inside or just stuck to the outside, we took this and washed it about 3 times in PDS actually while it was under the microscope, and we couldn't get this whole area of high signal to budge at all. So we think that probably we have introduced nanotubes into cells, and not only that, but they've stayed wrapped in the peptide through their delivery into the cell, because if they hadn't stayed wrapped by the peptide, they would have aggregated, and when they aggregate, the fluorescence is quenched and we wouldn't get any signal at all. So we probably are delivering actually nanotubes to cells using EQL6E GRGDS. Of course, microscopy studies are not a substitute for quantitative toxicity studies. So we did do a toxicity study with every surfactant. We did with the 2 chloronics, F68 and F127, and also with 2 peptides, EQL6E and EQL6E GRGDS, as well as SDDS. And we tested the toxicity of those surfactants both alone and also with nanotubes. And so this actually offered an interesting opportunity for us to assess the cytotoxicity of single-walled carbon nanotubes. And so, as you can see, at 150 micromolar, this is the final concentration of surfactant. None of these surfactants are appreciably different than each other. However, as we increased the concentration of surfactant, it became apparent to us that there were some differences in toxicity. Once we reached 300 micromolar, the SDDS population, their cell viability has now dropped to 0, whereas the other populations with the chloronics and the peptides have remained mostly viable, and it turns out that these guys are actually not statistically significantly different from one another. Furthermore, we can see that if you look at the striped bars with no nanotubes, so this is surfactant alone, and also the blue bars with nanotubes, the toxicity difference is also not different. And so this was an opportunity for us to assess the toxicity of nanotubes, and we had to come to the conclusion that at least to these fibroblasts, that single-walled carbon nanotubes were not toxic. So just to summarize this section, multidomain peptides self-assemble in the presence of single-walled carbon nanotubes. They allow better fluorescence than the chloronic series with comparable biocompatibility, and they also provided a great avenue to assess nanotube cytotoxicity. The next application I want to talk about is to use multidomain peptide hydrogels as a delivery agent for stem cell secretome. This is a project that we did in collaboration with Baylor College of Medicine, and specifically with Yin Wei and Farad Dinesh in the Department of Nephrology. Now, you guys have probably heard a lot of hype about embryonic stem cells. Embryonic stem cells are pluripotent, which means that they're a virtually unlimited supply of cells. They can differentiate into any tissue type, and they can secrete a variety of growth factors, cytokines, and other really cool compounds. And for this reason, they've been dubbed sort of the fountain of youth. And so they're very effective for use in emerging therapies. However, there are some drawbacks body, they'll be recognized by your immune system and promptly killed off. Even if that doesn't happen, you face engraftment issues, which means when you inject stem cells into your body, they may decide to settle in a place that you don't want them to settle in. Even if that didn't happen, they may settle down in a place you do want or a place you don't want and form teratomas, which are some really nasty tumors with a variety of different tissue types in them. There have been teratomas extracted from people containing complete teeth and hair and fingernails, as well as heart and lung tissue, all at the same time. So that's something we probably want to avoid as well. And then even if we were to avoid all three of those issues, you still face cell viability issues once the cells reach their destination. If that destination happens to be a diseased tissue or a tissue that's mostly dead, it's not really a hospitable environment for the cells. So you don't know that those cells are actually going to be able to survive in their targeted environment. So what we would like to do is harness all the benefits of stem cells without actually using stem cells. So using stem cells to make a stem cell-free therapy. And we think we can do that. And so the setup that we have is to take a culture of embryonic stem cells and incubate them in a cell culture well, where the cells are actually at the top of the well in a tray, where this is separated from the culture medium by a semi-permeable membrane. And so what happens is the stem cells are living in this tray. They secrete all kinds of great cytokines and growth factors, which diffuse through the semi-permeable membrane, down through the cell culture medium, and into a waiting peptide hydrogel that's at the bottom of the well. What we can do is just pull this culture of stem cells out, pull the peptide hydrogel out that's loaded up with all of these great cytokines and growth factors, and then take that cell-free peptide hydrogel, and we can either put that into another in vitro experiment where that peptide hydrogel will release those compounds back up into the cell culture medium where they affect another population of cells, or we can take that gel and inject it directly into a mouse where we can study a variety of effects. So I'm going to talk about both some in vitro work and some in vivo work. Our in vivo work is incomplete, so I'm going to talk more about the effects of the in vitro work, and what we're actually using that for is to treat a model of diabetes. Now the peptide that we have chosen for this mission is E2SL6E2GRGDS. So as we saw before, it forms great nanofibers, makes a great hydrogel, and is overall really nice for biological applications. And so our in vitro setup looks pretty familiar. We have our culture of stem cells. They secrete cytokines and growth factors. These are absorbed by the peptide gel. And then what we do is we take that peptide gel out of the cell culture well and put that into a new well that contains kidney endothelial cells. Now, kidney endothelial cells are important in diabetes because when the kidney cells are exposed to high glucose conditions, which is not infrequent in a diabetic state, they become leaky. And so this is the mechanism by which diabetics experience kidney problems. Their kidneys don't filter the way they should. And it's because these cells are a lot leakier than they're supposed to be. So when we took this peptide gel and exposed it to endothelial cells, we actually saw, well, we saw not much effect at an hour, but at 5 hours, we see a huge difference in the leakiness. So that's the relative permeability of these cells. Now, Hg is high glucose, and you can see that in high glucose conditions, these cells are a whole lot more leaky than in low glucose or normal conditions. However, then when we take our nanofibers, which are preconditioned with embryonic stem cells, so this third bar, we see a statistically significant decrease in the relative permeability of this endothelial cell population. And so that says that we have actually gone one step toward repairing these damaged kidney cells when they're exposed to this peptide hydrogel. Now, as a control, we also expose these endothelial cells to a peptide hydrogel that was preconditioned with cells that are not stem cells, so just fibroblasts. So MEF are fibroblasts and NF as nanofibers. You can see that when we precondition with something other than stem cells, the effect is not as dramatic. And so this says that we actually have partially repaired these damaged cells in vitro. before we can move on to some in vivo studies, we actually had to decide how this material was going to behave during drug delivery. And so we did a couple of rheological tests. The first was a strain sweep, and you can see that we have a G prime of around 475 pascals, which is a nice rigid hydrogel. It won't fall apart when we handle it. The second test we did was a strain sweep, or excuse me, not a strain sweep, a shear recovery test. And for this, what we did was we actually exposed the peptide to some low shear conditions here, and then introduced a shearing event. And this shearing event was meant to mimic the actual shearing that would happen when you loaded this into a syringe and needle and pushed it through as you injected it into a mouse or a patient. And so what we did is we did a complete shearing event, which means we totally destroyed the hydrogel. We know we totally destroyed the gel nature because the G prime at this point during the shearing event is lower than the G double prime. So we destroyed the gel. However, when we remove the shearing event, we see 75% recovery almost immediately and 100% recovery within about 15 minutes. And so what this test says to us is that when we push this hydrogel through the needle, we actually liquefy the gel, and then as soon as it comes out of the needle on the other side, it regains its gel structure. And so this is a great thing because we don't want to liquefy the gel within the needle and then have it floating around in the abdomen in a totally liquid state. We actually want it to resolidify on the other end so that we can do targeted therapies. And so this looked pretty promising. However, rheological tests are not a substitute for actual in vivo tests of this material. And so what we did is we actually decided to inject the peptide hydrogel into mice and track it by MRI to see if it actually would localize in the abdomen. And so MRI is great for tracking changes in soft tissue, but good resolution requires the use of a contrast agent. Gadolinium is the most frequently used contrast agent for MRI. And so what we did is we actually covalently attached a chelating agent to the N-terminus of our peptide, threw in some gadolinium, and this whole complex is irreversibly bound to each other, and this whole complex is MRI trackable. So we took our peptide hydrogel, labeled it, took the gel, loaded it into a syringe, and then injected that into a mouse. When we looked at the MRI data of the mouse, you can see that at 0 hours, so directly after injection, we have the hydrogel localized in this area here in the abdomen. At 3 hours and 6 hours, you may not be able to see it, but we actually still have those same points in red. After 6 hours, we have to wake the mouse up and reposition them, put them back to sleep later. At 24 hours, though, we still see that we actually do have a couple areas of localization here. And then if we take a higher resolution image at 24 hours, we can see that we actually still have an area of very high gadolinium localization in this mouse. And so when we did the initial injection, we were aiming for the kidney area, and 24 hours later, we still have a lot of hydrogel in that same area. So just to summarize this application, multidomain peptides form hydrogels that can absorb and secrete stem cell-derived compounds. These hydrogels recover very quickly after shearing events and therefore make up some pretty ideal injectable materials. They remain localized after injection of peptide hydrogel in a mouse. And given our in vitro results, we think that this could result in therapy for diabetes. Now just to go over some of the bigger conclusions from this work, multidomain peptides adopt a defined secondary structure at neutral pH based on their sequence. Substitution of different amino acids in the peptide results in structural and morphological changes. We can actually improve the rheological properties of the resulting peptide hydrogels through sequence modifications or enzymatic modifications. And then we explored 2 different applications. The first was that multidomain peptides can effectively solubilize single-walled carbon nanotubes for delivery to cells. And the second is we found that they can also be used as cytokine delivery agents, both in vitro and possibly in vivo. So with that, I'd like to acknowledge my committee, first and foremost, Dr. Jeff Hartgring, also Dr. Wilson and Dr. Tao. Everybody in the Hartgring Lab, thank you so much. Leslie, Jorge, Amshek, Marcy, Benoit, Brennan, and Nicole. And I'd also like to thank our collaborators, Dr. Bruce Wiseman and Dr. Demon Cybulski, Dr. Farad Dinesh and Dr. Yin Wang at Baylor College of Medicine, as well as my wonderful husband, Eric Bakota, and my parents, Gary and Adrian Floor. And my funding comes from the Welch Foundation, and for 2 years I was also supported under an NIH IBB biotechnology training grant. Thank you.